Okay, in part 2 of the endocrine discussion, we're going to finish off the worksheet. So let's look at number 4. It wants us to label these things in the thyroid hormone pathway. So the first integrating center, that's always going to be the hypothalamus. No matter which pathway you're in, the first integrating center is going to be the hypothalamus. And the final integrating center, well, that's specific to which pathway you're in. In this case, it was the thyroid. But it could be the adrenal cortex, or it could be the gonads, something like that. Whatever the final gland is in the pathway. The first efferent, so we didn't really talk about efferents versus afferents in the last video, but efferent, the way I remember the difference is efferents exit the organ, and afferents approach. So line up the first letter of these and you get the kind of mnemonic or trick to memorizing them. So the first efferent is the first thing that exits the first integrating center. So what exits the hypothalamus? Well, that's TRH. And the final efferent is the final thing that exits the final integrating center. So what exits the thyroid? T4. Number 5 is talking about the thyroid hormones T3 and T4. So if you remember from the homeostasis lecture, when Ian talked about T3 and T4, he said they are steroids, or at least they act like steroids. They are technically a single amino acid. However, that amino acid that they are made of happens to be hydrophobic, so these hormones act as if they were steroid hormones. Now, thinking back to the homeostasis lecture and what we know about a steroid hormone is that they can't be pre-packaged. They can't be pre-made and stored inside a vesicle in the cell. The reason why they can't be stored is because they are lipophilic, meaning they can diffuse into a cell membrane. So if you try to store them inside of a cell, well, they'll just move right out. It's impossible to store them, at least on their own. It's impossible to store them because they will diffuse out because their concentration gradient is moving out of the cell. So given that, let's read what this problem is saying. Well, it's saying the hormones T3 and T4, they're synthesized while attached to this protein called thyroglobulin. And just before they are released from the cells, they are stripped from that protein and they become lipophilic. Okay, so what it's saying is that even though T3 and T4 act as steroids, they are prepackaged, which goes against what we just learned, right? We just learned that, okay, steroid hormones in general, they cannot be stored. At least on their own, they cannot be stored. So let's think about that. But let's read read part A and see what it's asking. The first thing it's asking is, what is the mode of transport of T3 and T4? It's going to be simple diffusion. And then B asks, how does thyroid binding globulin function? So it's asking, what is the function of this protein? What is the function of it? Okay, so it's not asking like what is the biochemical mechanism of how these two bind together. So your answer is not a mechanism here. It's asking what is the function of it? Like what is the purpose of having that protein attached to T3 and T4 before the release of them? And if this is a protein, well, that protein is hydrophilic or lipophobic. So that protein cannot cross the cell membrane. And if that protein is attached to the steroid hormones, then that renders the steroid hormones lipophobic. So as they are attached to this protein, thyroglobulin, they cannot cross the cell membrane. So that function of the protein is to trap T3 and T4 inside until they are ready to be released, until they get the signal to be released. And you should know what the signal is to release T3 and T4. It is TSH. Once the thyroid gland gets the signal from TSH to release T3 and T4, it will clip off that thyroglobulin protein, which makes T3 and T4 hydrophobic and lipophilic, and then it can diffuse out of the cell. But the function of the protein is to trap or store them inside the cell until they are ready to be released. And then the last part says, give a teleological reason for why T3 and T4 have a nuclear receptor. You need to know what teleological means here. It is the why. Why would T3 and T4 have a nuclear receptor? Because they can diffuse past cell membrane. Basically because of simple diffusion. That's why they have a nuclear receptor. Nuclear receptor meaning it's inside the nucleus. So this is also something we talked about in the homeostasis lecture, kind of the general functions of a steroid hormone versus a protein hormone. A steroid hormone is likely going to have its receptor be found on the DNA itself, meaning that these steroid hormones are going to act as transcription and translation factors. So this is a little bit of a review from kind of genetics and intro bio. So transcription and translation, the goal of that is new protein synthesis. Right? You take DNA, turn it into RNA, and then turn RNA into amino acids, which you then string together to make a protein. So if these steroid hormones are acting as transcription and translation factors, their overall function is new protein synthesis. So let's use that knowledge to answer number 6. What is more likely the effect of T3 and T4 to be? Is it A, new protein synthesis, or B, upregulation of existing proteins? That's basically what those two statements are saying. And based on what I just talked about, well, it's the new protein synthesis, right? Because they are transcription and translation factors, they are going to stimulate the production of new sodium and potassium subunits. Sodium, potassium being a cell membrane protein. Now, to get this question right, you didn't need to know what the exact function of T3 and T4 are. And in fact, we don't expect you to know that. We don't expect you to know that T3 and T4 make new sodium and potassium pumps. But what we do expect you to do is to be able to apply the information that a steroid hormone stimulates the production of new proteins. Now, if we tell you that in a scenario that is specific to sodium potassium pumps, well, that's fine, because you're still looking for the word stimulate production. Answer choice B is referring to a protein hormone, upregulate sodium channels. Upregulation means you just send the existing sodium channels to the cell membrane. You're not actually making any new ones. You're just moving the existing ones onto the cell membrane. Also, IP3 is a second messenger, and you should know from the homeostasis lecture that protein hormones use second messengers, steroid hormones do not. At least in this class, at least for simplification. Steroid hormones do not use second messengers. Okay, let's move on to number 7, and this will be the last problem here. Okay, so number 7 has a graph, and whenever you're looking at a graph, you always need to understand what is being said on the X and Y axes. So the X-axis is concentration of T4. So the way you interpret the X-axis is if you are moving from left to right, you are increasing concentration of T4. That's what it's saying. And then the Y-axis is concentration of TSH. Meaning if you go from down to up on this, you are going to increase the concentration of TSH. In the middle here is about an equal concentration of both, or normal conditions, potentially. All right, so let's look at what the question is saying. Part A asks, what symbol represents data from a hyperthyroid condition? So the circle, I'm going to erase this stuff so we can see it better. The circle, the squares, or the triangle? Which one is hyperthyroid? Okay, so we have to think about in the pathway, in this pathway, which one is the thyroid hormone? Well, it's T4, because T4 is coming from the thyroid. So it wants to know which one is hyperthyroid, which one has a high T4. Well, like I said, when you're interpreting this graph, if you're moving from left to right, you're increasing T4. So the circles have a high T4. Okay, and then do the data indicate primary or secondary? All right, and this is where you just ask yourself, well, is T4 the last hormone in the pathway? Is it below that line? The question is yes, or the answer is yes, so it's a primary. So primary hypersecretion, because T4 was the last hormone in the pathway. So that one was pretty straightforward. All right, number 8, I'm not going to necessarily do because this is what I started part 1 with. So you can do it, but it's just like sensory signal to hypothalamus to integrating center 1, which releases hormone 1, that sort of thing. So go ahead and do number 8 on your own. It's exactly what I started part 1 with though, which is this. Okay, and then number 9 down here, you don't necessarily need to do, but I will talk a little bit about it. Number 9 is asking you to propose one mechanism by which thyroid and growth hormone could be permissive to each other. We're not necessarily going to ask you things about like make up a way that these are interacting with each other, but instead I would just recommend reviewing the 3 interactions: permissiveness, synergism, and antagonism here. So don't worry about number 9, just go review what those 3 interactions are: permissiveness, synergism, and antagonism. All right, so part 2 of this video was a little bit shorter than part 1. That's because the second half of this discussion is pretty straightforward, but these are still really, really important concepts for exam 2 because they tie homeostasis together into the endocrine system, and these will be things we're talking about all semester. All right, I will see y'all in Q&A hours some point this week or in your exam review if you scheduled a meeting with me.